Pharmapal.
Maternal environment influences the biochemical properties of Anastatica hierochuntica. Welcome to our exploration of how the maternal environment influences the biochemical properties of dead pericarps in the desert plant Anastatica hierochuntica. This study delved into the effects of abiotic stressors, specifically salt and heat, on seed germination and the biochemical composition of these pericarps. By analyzing phytohormones and metabolites, they uncovered fascinating insights into plant resilience and adaptation. Plants face extreme environmental challenges in the arid landscapes where Anastatica hierochuntica thrives. This study investigates how these conditions affect the seed's protective structures, the pericarps, during their development. They aim to understand the relationship between stress exposure and the biochemical changes in these dead pericarps, which play a crucial role in seed survival and germination. Materials and methods. To examine the impact of abiotic stress, they subjected A. hierochuntica to salt and combined salt-heat treatments. They meticulously extracted phytohormones and metabolites from the pericarps using advanced techniques like LCMS/MS and GCMS. Sample preparation. Plant tissues were ground in liquid nitrogen. Phytohormones were extracted with ethyl acetate and analyzed for concentrations. Statistical analysis. They employed unpaired t-tests to assess differences between control and treated groups, ensuring our findings were statistically robust. Results. Our results revealed significant changes in the biochemical composition of pericarps from stress-treated plants. Notably, they observed 1. Seed abortion rates. Control plants showed a 2.5% abortion rate, while salt and salt-heat treatments resulted in 8.3% and 12.9%, respectively. 2. Germination rates. Seeds from control plants germinated at 93% within 24 hours, compared to only 60% for those from stress plants. 3. Phytohormone levels. There was a marked reduction in key phytohormones like abscisic acid, ABA, and indole acetic acid, IAA, in pericarps from salt-treated plants. These findings highlight the critical role of the maternal environment in shaping seed viability and resilience. #discussion. The implications of our findings are profound. The increased seed abortion and reduced germination rates under stress suggest that A. hierochuntica has developed adaptive strategies to cope with harsh conditions. Biochemical adaptations. The alterations in phytohormone levels indicate a shift in metabolic priorities, likely aimed at enhancing stress tolerance. Ecological relevance. Understanding these mechanisms sheds light on the survival strategies of desert plants and informs agricultural practices in similar environments. We'll discuss the paper's key figures and tables, breaking down complex concepts into easily digestible pieces. Let's get started. Figure 1. Effects of stress on germination. Figure 1 presents the impact of stress on the germination rates of Anastatica hierochuntica seeds. Panel A shows the average number of control and stress plants that produce pods. Panel B illustrates the total seeds, including normal and aborted ones. Panel C highlights the percentage of seed abortion, revealing that stress conditions led to a significant increase in seed abortion rates. The results indicate that while the number of pods remained consistent, the stress conditions, salt and heat, negatively impacted seed viability. Figure 2. Germination rates over time. In Figure 2 is a detailed timeline of germination rates. Panel D tracks the percentage of seeds that germinated within 24 hours. Panel E shows the germination rate over time, emphasizing that seeds from control plants germinated significantly faster than those from stress plants. This figure clearly demonstrates the detrimental effects of stress on seed germination, with control seeds germinating at a rate of 93%, compared to only about 60% for stressed seeds. Figure 4. Analysis of hydrolase activities. Moving on to Figure 4, this figure examines the hydrolase activities in pericarps derived from stress-treated plants. The in-gel assays for nucleases, kinases, and proteases are depicted here, showcasing how these enzymes vary between control and stressed conditions. This analysis is crucial as it highlights the biochemical changes that occur in response to environmental stress, which can affect seed development and germination. Proteome analysis overview. Now, let's shift our focus to the proteome analysis section. 1. Sample preparation. The study utilized intact seeds and ground pericarps, incubated in PBS and centrifuge to collect supernatants for analysis. 2. Mass spectrometry. Proteins were digested and analyzed using LC-MS/MS, allowing for detailed identification and quantification. This rigorous methodology ensures that the data collected is robust and reliable, providing insights into the protein composition of the seeds and pericarps. Table 1. Differential protein expression. Next, we have Table 1, which summarizes the differential protein expression between salt-treated and control plants. Proteins are listed along with their respective fold changes and functional categories. Notably, proteins involved in stress responses, such as heat shock proteins and enzymes related to metabolism, are highlighted. This table is essential for understanding how environmental stress influences protein expression, which in turn affects plant resilience and seed viability. Statistical analysis. The statistical analysis performed in this study is noteworthy. An unpaired t-test was utilized to compare means between groups. The results were deemed statistically significant with a p-value of less than 0.05, indicating that the observed differences are unlikely due to chance. This rigorous statistical approach strengthens the conclusions drawn from the data. What are the main findings regarding phytohormone levels in pericarps from salt-treated plants? The study found a significant reduction in key phytohormones, particularly abscisic acid, ABA, and indole acetic acid, IAA, in pericarps from salt-treated plants. This suggests that salt stress can alter hormonal balance, potentially affecting seed development and germination. How did the researchers assess the differential protein expression in pericarps? The authors employed a proteomic analysis using LC-MS/MS to identify proteins released from the pericarp. They filtered proteins based on specific criteria, such as having at least 2 non-zero replicates, and then performed statistical analyses to determine differentially present proteins under control versus salt stress conditions. What role do hydrolases play in the context of this study? Hydrolases, such as nucleases and kinases, were examined for their activity in pericarps. The results indicated a reduction in nuclease activity in pericarps from stress-treated plants, while protease activity increased under combined salt and heat stress, suggesting that maternal stress conditions can significantly influence enzyme activity and, consequently, seed viability. Why is the study of Anastatica hierochuntica particularly relevant in the context of climate change? This plant species is adapted to extreme desert environments, making it an excellent model for studying stress responses. Understanding how maternal stress conditions affect progeny can provide insights into the resilience of desert plant communities amidst climate change, which is crucial for conservation efforts. 5. What implications do the findings have for agricultural practices in arid regions? The findings suggest that maternal environmental conditions can significantly influence seed properties and stress responses. This knowledge could inform agricultural strategies, such as selecting or engineering crops that can better withstand salinity and heat, thereby enhancing food security in arid regions. Thank you for joining us today. If you found this video informative, please give it a thumbs up and subscribe for more plant biology and environmental science content. If you have any questions or comments, feel free to leave them below.
Pharmapal.